Hello, and a very warm welcome to everyone joining us for today's SelectScience Webinar, Flow Cytometry, Insight into the Standardization of Instrument Setup across Cytek systems. My name is Dora Wells, and I'll be moderating today's presentation. I'm delighted to be joined by today's speaker, Dr. Maria C. Jaimes, VP of Application Support at Cytek Biosciences. In this role, Maria oversees a variety of R&D-related functions and leads Cytek's Global Technical Applications Support team. After the presentations, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A session at any time during the webinar. You can submit your question at the speech bubble icon to the left of your screen. And now, without further delay, I'd like to hand over to Maria for today's presentation, and I'd like to thank her for presenting to us today. Please go ahead, Maria.
Thank you so much, Dora, for the kind introduction, and it's my pleasure to be here. I'm going to start sharing my screen. I hope everybody can see my screen. So hello again, my name is Maria Jaimes, and I oversee the application support team at Cytek, and I'm very excited today to be talking about standardization of instrument across Cytek instruments. It's been a question that has been asked very frequently in the last few months, and we thought it would be good to have a discussion and to share with you all the information that we have about this fascinating topic. So let's get to the meat. What we are trying to achieve with instrument standardization? So the reason we are having this conversation is because many of you have collaborators in different institutions that have an Aurora or Northern Lights, and you want to run the same assay, the same study at different sites, or within your same lab or core facility, you have more than one Aurora or Northern Lights. And the question is, can I get equivalent results when the same sample is run on these different instruments, cross sites or in the same site? And when we talk about equivalent results and we talk to our users, the questions and the needs, what they express, they are most interested at is, if I have a given assay where I can identify X number of populations, and I collect data across instruments, can I use the same analysis worksheet? Can I use the same template in our software, in third-party softwares, where the gates are valid for all the data sets? So you don't have to be making gating adjustments. You don't have to be recreating gating because the data from one instrument to another looks different. The other reason that we get, or the other question that we get, is like, I want to make sure that the frequencies of the populations in my assay are the same across the different instruments, that I'm not going to get any bias or any discrepancies based on the instrument that is used. Another aspect that is mentioned is we want to make sure that the MFI of the populations is equivalent, which falls a little bit under the category that we want to use the same gates across all our files. And then the other aspect of it is I want to make sure that if in my assay I have dim markers, I am able to resolve them when I run that assay in any instrument, so that the resolution, the quality of the data is equivalent across instruments. So today we're going to learn and to dissect what is enabled in our technology to be able to meet all these requirements. So this is a very simple cartoon, and what I wanted to make sure is that we are all in agreement of what those questions mean. So if we have two instruments, instrument A at the top and instrument B at the bottom, what we want, if we have a positive, the green population, and a negative population, the orange one, and then a dim particle, the blue one, what we want to make sure is that the positives are in the same place in both instruments, that the resolution, the separation between the orange and the green is as similar as possible. Why? Because we want to be able to detect what is blue. And you're going to learn, it can be that the spread, how much spread you have of the negative is not identical, but that is good enough in both instruments to allow you to resolve the blue. So we're going to spend the first part of the talk talking about instrument setup, thinking, focusing on the positive part of the equation, the green population in this schematic. And later in the talk, we're going to tackle what is going on at the low end. So there are going to be 3 levels of standardization that we're going to discuss. First, standardization of setup using hard die bits, then standardization of setup with fluorescent particles. So these 2 are going to take care of that green population being at the same place. And then the third part is going to be looking at more in depth at the instrument performance, so the low end and the resolution. So first stop, and is the one where we're going to spend a little bit more time, is standardization of the instrument setup using hard die bits. So all our instrumentation our Northern Lights, the Aurora, and our sorter come with a built-in system, a strategy to achieve standardization of the setup using 2 components, our spectral flow QC bits, which we're calling hard die bits, and a software that has an algorithm that allows to have a consistent setup across time and across instruments. How does it work? First, what are these bits? These are hard die bits, our QC bits, are bits that fluoresce across all detectors, all channels in our instruments that are excited by every single laser that is in our instruments. They are not stained with actual fluorochrome. They have absorbed other dyes, but those dyes are excited by all the laser lines and have a very wide emission. And the commercial name of the bits that we use is spectral flow QC bits. Why do we like these bits? These bits are very, very stable over time across temperature. You can leave them outside. They will be fine. They're very robust. They give us very reproducible data across instruments and across sites, and they're easy to transfer from site to site because they're very stable. They're very easily available. You can order, we can get a lot of them, and they have very tight CVs, which is very important to achieve accuracy in the setup. On top of having these bits that we have fully characterized and that we feel very confident about, there is, as I mentioned, a next layer that is what the software does. So when you run daily QC, which I hope all of you are doing, the first user of the day, making sure the instrument is clean and running QC. When you run QC for every detector, there is an algorithm on the back when you run those bits that is making sure that the gains for each detectors are set so that the positive, the bits are put at the target MFI, and we're going to discuss in a bit how did we come up with those target MFIs. These target MFIs are known as Cytek settings, and every day the gains are adjusted to make sure that our QC bits hit those targets. And so what that means is that over time, we are going to make sure that our MFIs of any particle that we run within the same instrument are consistent, and that we have a consistent setup from instrument A to instrument B to instrument C, because we're using the same bits and the algorithm is placing them at the same target MFI. So why do we feel very strongly about having these target MFIs, and how were they established? So all of you who are Aurora or Northern Lights or CS users, you have heard and you have used and you have seen in our software the Cytek asset settings. The goal of having those settings as part of our software is providing you with a starting point to run your assets. And if you're running immunophenotyping assets specifically, we believe this is a very good place to start. I want to make clear, and I always say that, and I always get questions, do I have to use Cytek asset settings? You don't have to, They are not meant to be universal. They are not optimal for every application. For immunophenotyping, they are a very good place to start. You are always free to establish your own settings based on your needs, on the needs of your assets. And we have very clear guidelines on how to establish other settings. So be reassured, we are not telling you have to use those settings, but these are settings that we feel very strongly about for immunophenotyping and that can be further modified. How did we establish these settings? These settings are not random. There was a rationale behind those settings, and we spent a tremendous amount of time in the R&D team to get to a place where we were very happy. So we use biological samples, human cells, PBMCs or whole blood, to make sure that we were happy with those settings. We had to have settings that preserve the spectral characteristics of each dye, meaning that the dyes were peaking at the detector where the peak of emission is supposed to be, and that the spectral characteristics where you have those smooth spectrum signatures that you have seen in all our documentation. We need to make sure that we have optimal resolution so that in our settings we are in a place that if we run anything dim, you're going to see it. And we use anti-human CD4 initially to make sure that we have a good resolution. And we also use dim CD4s where you do a stain with a pure antibody mixed with a label antibody. So it's a competition experiment to make sure that very dim CD4 stain cells in this competition experiment were still detectable. We also make sure that at these settings we have enough range to accommodate bright signals so that we are not pushing the signal, the gain so high that if you run something bright, you're going to be off scale. And all these criteria so far are at the single stain level, but our settings are also meant, we also took into account a multicolor scenario because that's what the majority of you are interested at. And the settings allow us to minimize the spread, which is critical for multicolor applications. And we're going to talk a little bit of spread when we talk about performance. So all these criteria were taken into account. So as you can imagine, there was a lot of work to make sure we met all these criteria with the Cytek assay settings. Once we got those settings, we saved those settings using these QC beads to transfer the MFI targets of those settings across all the instruments. So if we did all this study in a couple of instruments, we ran, after we find the good place for the gains for every channel, we ran our QC beads, we established the target MFI of those QC beads, and that is the information that was transferred to the software. Then when you run QC and you run the QC beads, every day we're getting the beads in that target and the gains are automatically updated every single day. If you have your favorite settings, when you run QC, those are follow the same process as the Cytek assay settings, and they are also updated. So the same functionality and the same strategy around any user settings. So I have been telling you that we are using these beads, and there are various reasons why we are using these beads. It's a matter of being practical, ease of use. So the hard dye beads are very stable. They are cheap. They are robust. They are, again and again, we can see them in every detector, excited by every laser. And we found there's already a built-in automated way to achieve setup across our instrument because of the strategy and the algorithm in the software. What is their limitation? We are not saying they are perfect, and we know that there are limitations. They are not spectrally matched to fluorochrome labeled particles. So as I explained, these are not beads that are stained with the actual fluorochromes. So their spectra are different, very different to the actual fluorochromes that you run in your assays. And the fact that there is a spectral mismatch means that when you are going to run fluorochrome stained particles, you're going to see a little bit more variation in the setup because there is a mismatch in the spectrum. So they are not perfect, but they have great advantages and they provide us a practical way to achieve a consistent setup across instruments. So let's look at the data and see if what I'm saying is true. This is data from 4 instruments, 5 laser auroras, where we collect the QC beads at Cytek access settings, and we are looking in the y-axis at MFI, the x-axis are the different detectors, and you're going to see that there is a very high level of consistency in the MFI across detectors, across the 4 instruments. So they are very, very close. If we look at the CVs, the variation in those MFIs across those 4 systems, our CVs are all below 5%, some great majority between 2 and 3%. So we are able to achieve a high level of precision in the setup using those beads. Now, what happened when in those 4 instruments, I go and run fluorochrome stain particles? So here is the data. So these in the x-axis, what I have is a variety of dyes that are excited by all the lasers in those 5 laser systems. So we're covering all lasers, all wavelengths of emission. And here you can see the MFI of those stain particles. These are compensation beads. And you're going to see across for every fluorochrome that we test that there is variation in the MFI of those particles. How much variation? So as you can see, for some dyes, we have a good level of agreement. For some, not that great. So if I look at the CVs now, you're going to see that there's more variation. So the CV is in the y-axis, the dyes in the x-axis. And if I put a cutoff at the 10% mark, you're going to see that a great majority of the dyes are below 10%, but we have few that are up there. Some have higher level of consistency with CVs very low, around 2, but some have CVs almost around 14. So was this expected? Yes, because we didn't use these actual fluorochromes to set up the system, and we knew that there was a spectral mismatch. So if we look at the worst offenders, those that have a CV below above 10%, and we look at what happened in these 4 instruments, you're going to see that the spectrum, if we normalize the spectrum signatures are identical across those instruments, looking at the dyes where the MFI was very different. So the instruments are seeing exactly the same, but the MFI is not exactly the same because of the way we are setting up the instrument. So we have talked right now about the level of precision that we get with those beads, the QC beads, and now what we want to show you is how close can we get in terms of results of an immunophenotyping assay. So using these hard dye beads as our standardization particle. These data that I'm going to show you is part of the study that has been led by 2 of our technical application specialists, Chris Fleming and Amit Dankar. They have been doing an amazing job taking a deep dive at standardization of this assay across sites. I'm going to show you data on an 18-color immunophenotyping panel that we have as a lyophilized kit that we use as part of our training. So many of you have seen this data. And we have for this presentation, we are going to discuss 3 sets of data. What we call our gold standard is independent runs from the same lot of this kit on a single unit run by the very same operator. So this is what is our baseline, how much variation there is just by running the same sample over and over and over. Then the very interesting data set for us is what happened when you control as best as you can the sample. So it's a lyophilized sample, and we are very careful on how we resuspend and acquire that sample. And we're going to see data from 9 different sites with that same kit following a very strict protocol for acquisition. Then we were very interested what happened if we relax a little bit this protocol for resuspension and acquisition, and we're going to show you data from more sites showing you the impact of relaxing a little bit what happened at the sample level. So the second data set allows to clearly focus on the contribution of the instrument across in the variation across site. The third one allows to see the impact of the sample. So why using lyophilized cells when we have discussed this data? Sometimes some of our users have said, hey, but that's not a real case scenario. Well, the question is to be able to fully characterize and understand the contribution of the instrument in any variation that you can see in your results. So if we use a sample that is very variable, that has variability because the staining conditions, because of the sample, we're going to introduce more variation to what we're going to measure. So what we want to do is to focus on the instrument setup and focus on what is the contribution of the instrument itself on any of the performance results and reproducible. So we're using exactly the same biological sample. We are using exactly the same reagents and buffers to prepare that sample. We know that this sample, because we have done studies, is stable over time and over different conditions. Is a sample that is easy to ship across sites and is not going to have any decay in performance. Is a sample that allows to evaluate different levels of expression and population frequency and allows to minimize the operator contribution to the output. So many reasons why this is the ideal sample to do this kind of studies. This is the panel. As an 18-color panel. This, for you who are very familiar with our panel design materials, you know that this is the way that we like to present our panels. And here you have, it's a broad immunophenotyping panel. You can see we have markers for B cells, T cells, NK cells. So those are the main subsets, and monocytes. And I'm going to show you, using this assay, the gating strategy. So this is a busy slide, but very quickly we get on singlets, live cells from the monocytes. We can subset them based on some of the markers that we have. Then on the lymphocytes, we can look at CD3 versus TCR gamma delta, and we can subset the gamma delta T cells. We can also look out of the CD3 positive at NKT cells and different subsets of CD4s and CD8 cells that are here, and a variety of subset of, and we can subset the NK cells and the B cells. So here with the stars are some populations that are part of different cell subsets and that are identified with different subset of markers. And we are going to analyze what happened with those populations across the sites when we run exactly the same lot of this lyophilized kit. So here is I'm going to show you data of 3 sites: site 1, site 2, and what we call our reference site is a site where we had a high number of runs, where Chris have the user graciously gave Chris access to the instrument for several days. So this is one of the populations that we're following. That is the NK cells, 56 positive, 16 positive. And as you can see, by using exactly the same gate without moving, we're able to fit this population falls exactly in the same place across these 3 sites and across time. And we go with the other populations that we identify. These are the B cells. This is a subset of CD4 cells, another subset of CD4 cells, Tregs, and 2 different subsets of CD8 cells. So if you go column by column, what you're going to see is the same patterns, and that this gate at exactly the same position is able to fit all the data sets. So it was impossible for me to have a clear slide showing you plots from every site that we have collected data. But trust me, we can use exactly the same analysis template to fit the data coming from all the different sites that we have been using for this study. So now, if we look at the population frequency that is identified, and again, we're using some of the populations, we're not tracking all of them, but if we look at some of the populations. So in these next slides, in blue is our gold standard. So as I explained, the same instrument, the same operator, just telling us how much spread we get if we run the same assay over and over and over in the same instrument. Then in green is our very interesting data set where we have everything under control, the sample resuspension, the time of the acquisition, and where this is blue, data in green is going to show us the contribution from the instrument. And then the third data set in purple is what happened if we relax the conditions of the resuspension and the acquisition. For example, what happened if I resuspend the sample now and I run in 2 hours and another site running in 1 hour, and the buffer for resuspension is slightly different. So that is what that difference is. As you can see, the data in green is extremely tight and very close to the data in blue that is our gold standard. And as we relax the conditions on the sample end, that distribution start to widen, which is expected. So the green allows to focus on exactly what's the contribution of the instrument, and the purple shows that minimum variations on the sample end are going to show you bigger distribution, but it's not because of the instrument, it's a contribution of the sample. So if we look at MFI, at CVs for the different frequencies, you're going to see that the CVs for the gold standard are very tight, and that the bars in green follow very closely those CVs for the data in blue, which for us was like such a rewarding finding and something that we expected, but it's good to have the confirmation that our setup using our QC bits is giving us very reproducible results across sites when we use exactly the same assay. And as expected, as you relax a little bit the conditions in the sample end, those CVs start to get higher again. This is confirming everything that we knew from a theoretical perspective now with actual data. So this is frequency. Now we're going to look at MFI. So what happened with the MFI? Y-axis is MFI as our arc-seam transform scale, and then in the x-axis the different populations that we have been following, same color code, so the blue very tight, and you're going to see that yes, there is spread in the MFI as we go through more sites, and that spread gets more significant if we don't have a very tight control on what's happening at the sample end. These are the CVs. And you're going to see that the CVs across all the populations that we follow for the MFI are very, very tight. If we look at one instrument, when we open to more instruments, so again, focus on the bars in green, we are in average between, I would say, 3-4%, which to us is extremely good. It's a very tight set of data. Even if we relax the criteria on the sample, the consistency in the MFI is still good. So for us, for an immunophenotyping assay, these metrics and what the data indicates to us is that we have a very consistent setup. We did as well, and May and Chris are working on more detailed analysis that will be published, and part of it was presented at our recent user group meeting, and those presentations are available so on our website. So here, we're doing a dimensionality reduction using the UMAP algorithm, and we are overlaying the data from those 9 sites where the sample was very controlled. And as you can see, we have an amazing level of agreement between all those data sets in each site. We ran triplicates of the assay, so at the end we have 27 data sets here that match perfectly. Here, just to show you where are the different populations. So B cells are in yellow, is this island, for example. The TCR gamma delta are these cells here in pink, and our CD8 and CD4 subsets are here. So, sorry, I think CD8s are here, CD4s are here. So as you can see, the level of agreement, our ability to identify the same clusters is perfect when we run these the same assay, same sample, setting up the instrument with our QC bits. This is just to show you across operators, across sites, the triplicates, and how identical are those U maps, the separation between the islands. If you go through a column is a different site, you're going to see that the data is virtually identical. So we're very excited with this data. We are hoping to have a publication out of it soon. And we have a lot of data. We have a gold mine with this data that we have been collecting across sites worldwide. Okay, so we believe that the QC bits are doing their job and they are getting us where we need. So if you have an asset that requires a higher level of agreement, what I think is that maybe if you have an asset where you're tracking various changes in MFI, and then the level of consistency that you need for MFI is higher than what we show you, then the next step will be to standardize the setup of the instrument using fluorescent label particles. So why using fluorescent label particles? Because you are going to increase the accuracy by having exact match of the spectrum for the fluorochromes that are part of your specific assay. And this is very important. You cannot use a set of fluorochromes to standardize for assays that use another set of fluorochromes. The standardization has to be fluorochrome specific and for the specific fluorochromes that are part of the assay that you want to standardize. And with that level of standardization, then you're going to have the ability to compare changes in MFI for a given marker. So you're going to increase the level of precision of the measurements that you're doing. Unfortunately, there is a reason why not everybody's doing this and why it's hard to accomplish. So it's very difficult to find stable fluorochrome particles that are commercially available. If you do this in your lab and you stain compensation bits from any vendor and you run them at time 0, 1 hour, 2 hours, you're going to start seeing changes in the MFI. Even if you fix them, they are not super stable. So it's very challenging to find the level of stability that we have in the QC bits in fluorochrome label particles. Even if you find some that are commercially available, it's going to be, in my knowledge, and from everything that I know, is almost impossible to find them in all the fluorochromes that you need. So we have a beautiful instrument that allows you to run any possible fluorochrome and to find particles that are stable for to standardize every single fluorochrome is not possible at the moment. It becomes expensive because, again, the limitation on the particles that you need to use. It's very difficult to reproduce over time because the lot-to-lot variation is there. And it's very hard to get these lots to be consistent across time. And it takes more time. With the first method, as you heard, is part of the QC. You run your QC, you're good to go. Here, we are going to have to do some more work. So what do I mean by more work? And we have done everything in our power to make this as easy as possible if you are interested. So you are going to take one instrument, any instruments of the ones that you want to standardize, and you are going to run as part of the reference group all the fluorochromes that you are interested at standardizing. Then in that instrument, you are going to read what is the MFI, in this case for CFluor R668, which is the equivalent to APC. You're going to get what is the MFI in that instrument of those label bits. And then you're going to run the same particles on instrument B. And in this schematic, what you see is that in instrument B, the MFI is not exactly at the same place, as we already explained, because we are using the hard die bits to, at this moment, we have only used our QC, our Cytek assay settings to run these particles. So then, taking advantage of the beauty of our system, that is that there is a linear relationship between MFI and gain. If I know what is the MFI of these particles in instrument A, and in instrument B, I know what is the MFI that I'm getting, and I know what is the gain, then what I'm able to do is to do a simple mathematical calculation to calculate what is the gain that I need on instrument B to get those same particles to the target established in instrument A. And so these here looks very simple, and is simple from the mathematical perspective, but it's time consuming because you need to run all the particles on instrument A, then all the particles on instrument B, and then we need to do these calculations. We have an amazing template and a protocol that was established by our scientists, Huimin Gu and Alex Zong, and so we are able to help you and to streamline this as much as possible, but it does take time. I'm not going to tell you that this is a 5-minute affair. It's going to take some time, and the more fluorochromes you need to standardize, the more time it will get. Once you have done the exercise on each instrument that you're going to standardize, you're going to save those settings as standardization settings, and you're good to go. You're going to track their stability over time using the QC beads, but now every time that you run the same fluorochromes in each machine, you're going to reach a higher level of agreement. How much more agreement? So these are 4 instruments, again, dyes that are expanding through all the laser lines, all the wavelengths, and now you can see that the bars are about the same level across the 4 instruments, and you're going to be amazed. I left this access to 20%. You're going to tell me why you left that scale. Well, that was the scale of the first set of data, and we were having things around the 14%, if you remember. Now, everything is below 3%. Even below 2%, I could have been more stringent, but there is 1 or 2 that are a little bit above 2%. So the level of agreement that we can get at the fluorochrome level of the fluorochrome stain particle is significantly better. But again, there are some caveats, and it's not so easy to achieve these across sites. This is data acquiring 4 instruments, and now you can see we have, when we look at the CVs, they are way, way tighter because we did standardize the fluorochromes using fluorochromes stain particles. It's achievable, it's doable, and I think it has its merits if you have specific assets. If you are just doing immunophenotyping, I believe that our QC beads, the hard dye beads, can get you to a very good place. So we have talked at length and how to get that positive population to exactly the same place. So we cover this guy. How can we get this positive particle to be at the same place? Now we're going to go to the lower end. What happened here? How do I get same resolution? Okay, so for the positive signal, for the how much separation you get, the main contributions from an instrument perspective are in the optics. On the excitation end, the laser power, the laser wavelength, how good is the alignment. On the collection end, the quality of your optic and the sensitivity of your optical detectors, the filters that you have, how wide they are, how good is the transmission, and also the alignment. Yeah, I made a mistake here. It's not laser alignment, but the alignment of those optical elements that are part of the collection, in our case, in the APD modules. When we think about the negative side of the negative side of the equation, not the positive, but the negative, we have negative spread, the spread of the negative population measured as an RHD. That is the classical way that we measure in flow cytometry. And the 2 major components of that R&D are optical background that can come from your sheet, from the cleanliness of your instrument, the cleanliness of your flow cell, Raman scatter, any light scattering that is coming that is not due to any particle that you're running, and the noise coming from the electronics of the system. If you have heard our talks, you know that we made a huge effort in the design of our instruments to get the electronics as quiet as possible. It's something that we are very proud, but there's always going to be some level of electronic noise. It's impossible that it's like 0. There will always be something. So knowing this, in our manufacturing procedures, we have done everything in our power to make sure that the performance is as consistent as possible. From the laser point of view, we have specs around our laser power, our laser wavelength. We have a lot of specs around our optical filters, our specs around the APD module performance. Here I'm showing you what we do, a scan of our filters. We have specs by running hard dye beads, the MFI of some of these peaks. The second brightest peak and the second dimmest peak have very tight specs in terms of their CVs and their MFI. We have specs around electronic noise. So we go through a very lengthy process to ensure that the performance, optical performance, is as good as possible. How can you track that performance, that ability to resolve the dim population from the noise is there? Many of you, and this is classic, run multi-peak beads, 8-peak beads or 6-peak beads. We use as part of our manufacturing process and as part of our installation process 6-peak beads called Cubisure beads. At the top, I have instrument A, at the bottom, instrument B. And my eye, when I look at the Cubisure beads run in our system, is in the longer wavelengths for every laser, because at the middle, in the third or fourth channel, we have a lot of light and you are going to see all the peaks. But the question is, how far can I go at longer wavelengths until I cannot distinguish the two peaks? Here I'm showing you what to me is always my eye is. For the UV module, I need to make sure I can separate the two peaks around the 10 channel, that is around 610 nanometers. For the violet, for me at the 14 channel, with is 750 nanometers, I should be still able to distinguish the dimmest and the second dimmest peak. For the blue, around 760 nanometers as well. This is blue 12, I should be able to distinguish. And for the yellow, green, and the red, all the way to the last channel, you should be able to detect to differentiate the dimmest and the second dimmest peak. You're going to see that the distribution is not identical. There are differences from instrument to instrument. As much as we try and we put our specs in place, it's a complex system of optics and electronics, and it's not going to be possible to get exactly the same distribution at the bottom of the scale when you run these hard eye beads. So, however, the level of resolution should be there. Now, another layer is if we run fluorochrome labeled particles. And what we use is stain index. All of you who have been to our trainings and have been to any flow cytometry talk, you have heard about stain index. And here is a comparison across many dyes of stain index between those two instruments that we have been comparing. And you're going to see that the trend, if I'm going to find the highest and the lowest stain index, the distribution is identical between the two instruments, but the absolute number is not identical. Why? Where is that variation coming? Okay, from the positive end, we already know that the fluorochromes are not going to be at exactly the same point across the two instruments. So there are going to be variations in the MFI. And I just show you that at the negative end, if I look at RSD, they are not identical. They are very, very close. I hope that you can see and you can be mesmerized on how close they are, but there are areas where they are different. And because you're using the signal divided by the noise, you're going to end up with some differences. But in all these instruments, the question is, are you able to resolve the dim from the negative? And we are going to see, in my opinion, the answer is yes. I just want to quickly advertise a hidden gem in our report, in our QC report that is saved as a CSV file under the Cytek Bio export. We have a sensitivity metric. That is a metric that is generated during the QC, during your daily QC. We're dividing the signal of the QC beads by noise, and we have a way of calculating this. And you're going to see in this instrument, for example, in the yellow-green, that one instrument has a higher sensitivity. The higher sensitivity number, it means that you have more resolution. And no surprise, that metric correlates with what we see in the stain index, right? Because we're measuring the same thing, signal versus noise. Here, using fluorochrome labeled particles, here using our QC particles. So take a look at that sensitivity metric. If you have various instruments, it will help you to understand what differences in performance you have between the different units you're comparing. And now, if there could not be a talk about performance without talking about spread, our favorite topic. So these are slides that you have seen from our training materials, two fluorochromes, fluorochrome 1, fluorochrome 2, at the single stain level. We're very happy we can distinguish them. Now, when we mix them, we can have a very happy scenario where these two fluorochromes are not impacting each other, but we can have a more challenging scenario where one of the fluorochromes is spreading into the other one, and that spread is going to affect resolution. So if we're going to talk about instrument performance and standardization, then we need to talk about what happened in the multicolor scenario. There are different ways to measure spread. Cross-stain index and the spillover spread matrix are two. There is a publication for this. This is a metric that we use more at Cytek. Both are part of our training materials. Both have information about what happened in the multicolor scenario when you have spread from one dye going to another. I don't have the time to go into the detail of this, but there are two metrics that we use to assess spread across many dyes. When it comes to comparison across instruments, we have seen that the spillover spread matrix is more robust in the sense that it's less affected by changes in the brightness of the particles that we run. So because we're tracking data over time, we tend to favor the spillover spread value over the CSI, but you can use them both. So this is a beautiful table where we have many dyes and we are color coded, and this it's work from Alex Zong. And basically what we see here is that the areas in red are fluorochrome combinations where there is a lot of spread, and you have seen these in all our training materials. And when it comes to panel design, you're going to be very careful on what you do in these areas. So now the question is, this is Aurora A. How does things look in Aurora B? And you're going to see that in Aurora B, things look awfully similar. I'm going to toggle back and forth. You're going to see that the color codes and the areas where there's more spread and the areas where there's little spread are virtually identical. So then what we do when we're comparing spread is we subtract one matrix from the other. And this is what happened when we subtract them. Is color coded. Blue is when there is more, the number is higher on the instrument we're subtracting from. In this case, Aurora A, and pink is if there's more spread from the first item in the subtraction, so Aurora B. And you're going to see that there are areas where there is more pink, there are areas where there is more blue. All these numbers are, even if you are like, oh my God, what does she want us to look? Every number here is below 3. And that for a flow cytometer, if you look at SSM, is amazing. It means that the spread between the two instruments is very, very, very comparable. You can do the exercise with other instruments. I bet you are not going to get this level of agreement. If you see differences starting to be higher than 5, start taking a close look. Higher than 10, we need to investigate what's going on. So this is amazing. If you are familiar with SSM, you know that values higher than 10 are problematic. And here we're showing a high level of agreement in the spread between the two systems. And what does that mean? It means that when we want to identify dim populations, we don't have any problem, and the two instruments, and in this case, at 3 different sites, behave very similarly. And again, when it comes to resolution, we're keeping our eyes in the negative populations. Those populations are centered around 0, and they're very nicely clustered and easily to identify. If we look at T-Rex, same. If I have a distribution where I have high, bright, dim, and negative, I don't have any problem seeing those different levels of expression and identifying the cells of interest. Why? Because we have a robust process, a very smart design on our instrumentation, very tight specs, and we're able to offer you systems that have the same level of resolution. Very quickly, one thing is what we can do internally, another thing is what people can do in their labs. There's very little time left, but I just want to encourage you, for those of you who are doing multi-site comparisons, to take a look at this paper that was published last year in Bioanalysis by some of our users, and we have been working in the past few years very close to Q-square solutions. They have several of our instruments, and in that paper, What they did was they create their own user settings. There were things about the Cytek asset settings that they wanted to improve, and this is just across 120 days. They're tracking the stability of their system. This is one of their supplementary figures, and you're going to see that the data is very stable. Then they had 4 auroras. Seems like 4 is the magic number. And here you're going to see they're comparing a stain index. You can go row by row, or you can look at the CVs that they're getting. And surprise, surprise, very similar to the data that I showed you early on. So they're seeing the same. The stain indices are not identical. And by now, by the end of this talk, I hope that you understand why they're not identical. But they're very, very close. And then they show the metric that they chose to evaluate spread. So they listen and they really follow our advice was CSI, and they're showing the CSIs across the systems. You're going to see that the overall patterns where there is a lot of red is consistent across the system, and that there are some areas of discrepancy. Is this expected? Yes. Is this going to have an impact on your immunophenotyping assay? Very likely not. So, in summary, when it comes to standardization of the instrument setup, of the positive aspect of the equation, those positive populations, for immunophenotyping assay, we believe we have a wonderful built-in solution that is fast, that gives great accuracy, and that allows you to reuse worksheets. It allows you to reuse controls. We're very satisfied with the performance. If you have an assay that requires more precision in terms of changes in MFI, we have the tools available to you to do the setup with fluorochrome-based particles. The limitation that we have to overcome and that we have to brainstorm is what are the particles that we're going to use. When it comes to instrument performance, please do a good maintenance of your system. Be very good at cleaning. If you don't clean well, you're going to be penalized with the optical noise. And if the optical noise comes high, you're going to compromise your resolution. The performance at the single stain level is enabled by the manufacturing process. We have different metrics to track it. Multi-peak beads, stain index, sensitivity. We discussed these metrics. They're all available to you. We use many of these as part of our manufacturing process and installation at the multi-peak level. And stain index and sensitivity, you are already using it, or contact us if you want to learn more about it. And when you are going to be in the multicolor scenario, you know that spread is the main variable that we need to take into account. It's a key variable when it comes to panel design, and is a variable that we need to assess when it comes to compare instrument performance. We have a lot of tools available to you to do these comparisons, and we have guidelines on how to interpret the data. With this being said, I want to thank you all for attending today before the long weekend start for those in the US. I'm very grateful for your support and your interest about what we do at Cytek. And with this, I would like to open it for any questions. Thank you so much.
Fabulous. Thanks a lot for that interesting seminar, Maria. Now let's move on to the question and answer session. So the first question we've got here for you, Maria, is if we create our own settings, would it be a good idea to define them as delta from the CAS? So rather than using the same gains every time, define a gain as, say, -20 from that day's CAS.
So if you create your own user settings, I don't think there's a need to calculate the delta. I think if you need, if I understand correctly, if you are going to be tracking your own settings, you are just going to use the same functionality. You're going to make the adjustments that are needed, and then you're going to save those in the software with whatever name you want. And every time that you run QC, those settings are updated as the Cytek asset settings, and you're going to be able to consistently use them over time. If the question is how to optimize those settings, how to get to those optimal settings, it will take a little bit of time to explain. Basically, don't don't adjust individual gains. It's always a matter of proportions. But there's no need to do these adjustments on a daily basis. Basically, you do them once, you save them, and once you do it, when you do the adjustment, do them wisely. Consult with us if you want to understand how to make sure that those settings are optimal, and then you save them and you reuse them as you will reuse Cytek Access settings. So you do it one day and you're set. I hope that answered the question.
Great, yeah, thanks for clarifying that one, Maria. The next question I've got for you is, before you run sample to different site, does Cytek do any adjustment of the instrument?
So before you run a sample, so when the instrument is delivered and the instrument is installed, the engineers go through an installation process, and they will run many different tests to ensure that, as I showed, that the performance is comparable. Then the user has access to that report of what happened to the installation. If you are asking if you need to run any special tests before you're going to run a sample across multiple sites, then I will say that comparing the QC reports is a good idea, making sure that your instrument is clean. Comparing your QC reports, I will suggest that you contact our wonderful application support team so that we are sure that you're up to go. I will check probably the multi-peak bits to make sure that I have enough resolution in both systems to be compared. So I think, to me, we need to make sure that the performance of each system is as optimal as possible, that the instrument is clean, that you're passing QC with high marks, hopefully, and that you have a sense of the resolution of the systems. And then if you want to start a study that is really, really detailed and you want to have the granularity on how the two instruments perform, we can talk about doing some fluorochrome-based tests as I described in this presentation. So contact us, and depending on your needs, we can tell you which are the best tests to run before you start your study.
Great, thanks a lot for that, Maria. And we just have time for one or two more questions. And the first one is, the high consistency across sites is what we would like to achieve. So is it always achievable following the standardization protocol, even if samples acquisition is taking place over a longer period of time, such as 12 months?
So standardization is achievable. In this talk, I hope that I convinced you that we are able to minimize the contribution of the instrument to your results. So if you're going to run a cross-site study, longitudinal study, we are confident that with our instrument, we are able to give you consistent performance across time and across site. And that the variation that we are seeing in the results is actually due to the biology and not due to the instrument contribution. So do I believe it's achievable across time, across sites? Yes, I do. I think we have the tools and the capability in terms of the hardware and the software performance to achieve that.
Okay, great. Thanks a lot for that one. I think we have time to squeeze in one more question, and that is, can you import and use experiment templates between identically configured instruments?
Yep. Yes, you can, and it's super simple. Just export those templates. If you have an intranet or if you have a memory stick, whatever your facility allows you to use, and then bring it to the other instrument, and you can import. Make sure that your instruments or the computer analysis computers are using the same version of our software. We are currently under SpectraFlow 3.0. If you have older versions of the software, you can go from the lower version to the upper version, but not vice versa. You cannot go from 3 to 2.2. So just make sure of the software version. And yes, sharing experiment templates is as easy as a few clicks and you're done. And worksheet templates, you can also share settings. So there's a lot of functionality in the software that allow you to share experiments and templates across sites.
Great, thanks a lot for that. I'm afraid I'm going to have to wrap it up there, but thanks again to Maria for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I hope that you've all found this a worthwhile session. And if you have any other questions, please feel free to email me at editor@selectscience.net, and I'll follow up with your questions for Maria. Remember, you can also download a certificate of attendance in the Related Resources tab to the left of your screen. If you would like to listen again to today's webinar or invite a friend to listen, it'll be available to watch on demand in a few days. Goodbye, and thank you once again for joining us.